On today's episode,
humans are inestimable value, right? So we're products of divine intervention, created, we're loved by God, we're made in God's image, and all this gives us like this intrinsic value. And so because of that, we have a responsibility to protect and care for our own lives, stewardship of our own body, but also we have a responsibility to our neighbor to protect and care for the most vulnerable around us. And that's what makes bioethics surrounding vaccines complex. Because you have the one question of like, what's best to do for my body on a kind of a personal level, but you also have the question of what's best to do for my neighbor.
Welcome to the Life Challenges podcast from Christian Life Resources. Our world today presents people with complicated issues of life and death, marriage and family, health and science. It can be a struggle to understand or deal with them. We're here to help by bringing good information and a fresh biblical perspective to these matters and more. Join us now for Life Challenges.
hi, and welcome back. I'm Krista Potratz, and I'm here today with Pastors Bob Fleischman and Jeff Samuelson. And today we have a special guest with us, Dr. Rob Balza. And we are going to talk about the subject of vaccines today. Some of our maybe long-term listeners here will remember that you were one of our first guests on the podcast a few years back. It's great to have you back with us again here.
He's gotten so much older and wiser now.
Grayer, maybe. I did hear that was the highest rated podcast ever. Is that true, Krista, or is that just a rumor?
Yes, it is. But we're glad to have you back here. And last time I think we talked about a variety of different topics, but today we really wanted to focus on the vaccine topic. So can you just describe a little bit for our audience here, your background, specifically like kind of in talking about vaccines and your interest in this topic?
Sure. So I'll start by saying that I did for a very short time work at a pharmaceutical company. However, I'm not employed at a pharmaceutical company, nor do I invest in pharmaceuticals. I believe I have no conflicts of interest with this, with regard to vaccine development and things like that. So, and I also say, I'm mentioning things I'm not. I'm also not a physician. If I'm perceived as giving medical advice at any point today, consult with your physician and with regard to all medical decisions. But I am a biomedical research scientist. So I was trained at the Medical College of Wisconsin. My advanced degree is in biochemistry, but I do research with development and muscle and involves genetic studies and how proteins work. Right. So I work right at the molecular mechanisms of how our DNA, our genetic material, produce this wonderful molecule called messenger RNA and how that's turned into proteins that do all the things that our cells do and make us who we are. And that's often, the level at which diseases, often it boils down to things happening at the molecular and cellular level. So the things I study are the foundation for lots of disease processes and immunologic processes that involve vaccines and things like that. For over 20 years now, I've been teaching nursing students, pre-med students, and things like that at the college and university level. And now I do quite a bit of academic administration work.
Well, thank you so much for explaining that. For our listeners, especially maybe those that don't have a medical or science background, how would you explain what vaccines are and how they work?
Sure. So I'll do a little history lesson as well, kind of share, because I think if you think through kind of the history of vaccine development, it also kind of reveals how vaccines work on a practical level. But, vaccine technology isn't new, right? So if you look at history of science books, they'll trace it back to as early as the 15th century. Some people go all the way back to before Christ. There it gets a little sketchy in terms of what we know. But what's really clear is that in Europe in the 15th century, smallpox was a big deal. Back then, millions of people would die a year from smallpox. It had something like a 25% mortality rate. And interestingly, what was noticed over time is that if you took a little bit of pox from someone, it manifests with blisters on the skin. And if you're not driving right now when you're listening to this podcast, you can search, smallpox on your phone and you'll see pictures of people with these pox. And you can take a little bit of that and say on a knife or something like that, and you can scrape that into your skin. And typically what happens then is you do get kind of this little minor infection where it's rather than like getting down to your lungs and make you completely sick. You'll get little blisters and things like that. But that's enough to generate what's called an immune response. This technique became known as variolation, taking a little bit of pox from someone else and scraping on your skin. So now you didn't get the full-blown disease, but you triggered an immune response. So your body started attacking that virus or bacteria to protect you against it. And what they discovered is that mortality rate from someone that went through that procedure dropped from 25% all the way down to 1%. And odds of getting infected all went way down as well. And that technology, that's how it existed for something like 200 years, and it was passed down from word of mouth. There's lots of interesting stories about physicians traveling to foreign lands and seeing this practice and bringing it back and talking about it in their community and doing experiments with it. But it really wasn't until the late 1700s, there's a gentleman, an English physician by the name of Edward Jenner, who took that technology and really took a scientific approach to it and kind of formalized it. What he did is he discovered there's a related virus that infects cows called cowpox, appropriately enough. And milkmaids will sometimes get these kind of a lesser infection in their hands, these pox. And what he found is if you did a variolation with a cowpox infection instead, it has less severe side effects, right? But it also protects against that related virus, the smallpox virus. That's where you get the name vaccine. Vaxa, it means cow. Right. So, and this is, I consider this to be kind of the first real vaccination was then against smallpox. And I mentioned before, smallpox is killing millions of people per year. This is a big deal. This is the leading cause of death. The most historians will estimate something like 10% of people died from smallpox.
How did smallpox kill? What did it do?
It's infectious. It gets down in your lungs, gets into your bloodstream, spreads through your body and just kind of overwhelms your body, as many viruses do, right? Transmitted in ways similar to what we're all experiencing with COVID and flu and things like that. You can think of it that way. And now it's this historical note, right? And that's the most interesting thing of all is as of the 1980s, smallpox has been eradicated from the world. And that shows you how powerful vaccination is. So in the 60s and 70s, the World Health Organization had worldwide vaccination efforts. Countries got together on an international way for vaccines across the world, and we eradicated this virus. Interestingly, it does still exist in, some freezers in carefully guarded labs in two places, the United States and Russia. What could possibly go wrong with that? But what an amazing success story. You think about how many, hundreds of millions of lives has been saved since that virus has been eradicated due to vaccine. And then fast forward another 100 years, we're in the late 1800s, 1885, probably you've heard of Louis Pasteur, not a physician. He's a guy like me, he's a scientist, but he developed vaccines, for example, for typhoid. Typhoid is not caused by a virus, it's caused by a bacteria, but both viruses and bacteria, these tiny little things that what they have in common is they can, they're much, much smaller than our cells, right? And they can multiply very quickly and spread through your body and make you very, very sick. So typhoid was caused by Salmonella bacteria, which you still hear about sometimes, Salmonella typhi. It's a water spread bacteria. So sanitation improvements and different things like that, just washing your hands has greatly reduced the spread of so-called typhoid fever. But it's been estimated that Louis Pasteur's vaccine for typhoid saved something like 50,000 lives per year since its discovery. And then in the 1900s, the name that pops up is Jonas Salk. There's the Salk Institute, still developing vaccines and doing fundamental research here to this day. And what Jonas Salk contributed to vaccine development is he realized that rather than using a live virus like they were doing with variolation and Edward Jenner did with smallpox, you can use what's called an attenuated virus. And this is a really interesting thing. And I think a lot of people don't understand kind of how that works. But what he did for polio is he took the polio virus and he managed to grow it in culture, so in incubators, which is a creepy thing to grow up a pathogen in an incubator. And this is still being done all around the world. This is how we really understand how diseases work, right? So I've got a colleague, Jim Hinkle, shout out to Jim if he's listening. He grows chlamydia in petri dishes the same way. So it infects human cells. He grows little glass or plastic petri dishes filled with human cells, and he infects them with this chlamydia bacteria and watches the disease process and tries to find drugs to see how they work. This is what Jonas Salk did with polio. He grew human cells in a dish, infected them with this virus. And then interestingly, this is the really innovative thing. is he, over time, changed the conditions in which those cells were growing. So typically you would grow human cells in a petri dish in an environment that is very similar to the human body, right? So 98.6 degrees Fahrenheit or 37 Celsius, and then all the right vitamins and nutrients that you would have in your bloodstream feeding your cells typically. But what you can do is you can change those conditions. So, for example, one way you could do it is you could slowly, lower the temperature at which you're growing the cells. Go from 37 degrees C to 36, and then to 35, and then to 34. And what's interesting is that virus will adapt over time to that environment. This is evolution, right? Evolution on a small scale. The genes change, mutate, so that the proteins are better suited for those cooler temperatures. And so if you do that long enough, the virus adapts and grows really well at, say, 30 degrees Celsius. But then it doesn't survive well at body temperature. And so then you take that virus out of a 30 degree Celsius incubator and you inject it into a human, that virus doesn't do well. It starts, it kind of sputters a little bit and grows a little bit, enough to make some proteins and things, but you don't really get sick because it doesn't thrive at that temperature. And then your immune system can really kind of take over and attack it, produce these things called antibodies, which are these wonderful proteins that stick to the virus and keep it from infecting other cells and identify it so your immune cells can come gobble it up. But so that's what an attenuated virus is. It's a weakened virus that doesn't really make you sick, but is similar enough where it triggers an immune response. So that gives you then lasting protection against the real thing. So then when you encounter polio, native polio in the wild, your immune system already kind of knows what it looks like. You've got antibodies circulating in your body that protect you against polio. That's why you don't see too many people that are paralyzed with polio anymore. And back when Jonas Salk was working, right, in the 1950s, this is something that affected millions of people. American presidents, I can't remember which one, was paralyzed with polio very famously, right?
FDR.
Yeah, thank you, FDR. You can tell I'm not a historian. Trying to talk about history, and I'm a biologist. So a little outside of my comfort zone, but again, estimated to save millions of lives, prevent tens of millions of cases of paralysis. Absolutely incredible. That's kind of a brief history of kind of how vaccines work, and hopefully that gives you some idea of the biology and kind of the how we develop these over time.
Yeah, I just wanted to emphasize that the history there is important, and not just for understanding how vaccines came to be, but precisely because of the blessing that vaccination has been. So many Americans and people elsewhere in the world have no clue what it is that they've been protected from. You talk about smallpox. Well, that was a problem at some point. Polio, I think I remember hearing about that. Oh yeah, it was in American history class. There was something about FDR. Yeah. I am old enough that I know people who knew people who had suffered from polio infections and things like that, and things like the measles and German measles and rubella and some of those things that we've been vaccinated against. People just don't realize how bad those things were. And, it's the curse of being blessed in a way. And so this little historical thing is a good reminder to us, like, oh, things haven't always been the way they are. And maybe they could go back to that if we aren't careful.
It's interesting, too, because some of the skepticism and concern about vaccines, that's not new either, right? Mm-hmm. My wife has a grandmother that's in her late 90s. She's very sharp and still tells wonderful stories, and recently told us a story about how one day at school, she was given by the school nurse a polio vaccine. And she got home and told her dad, and her dad was furious and thought this was some suspicious government conspiracy and tried to scrub it out of her arm with a bar of soap in the bathtub. Wow. And I thought, man, some things never change, right? Yeah. So we're still dealing with those kinds of conspiracy theories and things like that surrounding vaccines too.
Yeah. Well, kind of in that regard, Rob, I think it was called the Cutter incident, where it was polio, but it wasn't attenuated fully, and it got out there. And the only reason I mention it is because sometimes critics of vaccine tend to pounce on where something goes wrong. And the attenuation is a pretty decent science. I mean, it's not like, hopefully it worked this time, but it's not that precarious. But people do recall some of that stuff, or at least they'll recall it from a history book.
Right. And you're right. It's not perfect. And the success in aggregate is overwhelming and awe-inspiring. These are some of the most successful biomedical inventions in all history. And then you think about the millions of lives that have been saved. But again, they're not perfect, right?
Mm-hmm.
So flu vaccine is a good example. Sometimes even in a good year, it might reduce your odds of getting the flu by 50%. And someone goes to the trouble of getting their flu shot and they still get the flu. And it's like, what in the world did I do that for? Obviously, that was a waste of time, and that's the last time they ever get the flu vaccine. Sometimes the vaccines don't work. Sometimes there's a bad batch. Sometimes it's injected incorrectly. There's a lot can go wrong. And sometimes you have a failure in the attenuation process, and someone can get sick, and that does happen. And it's funny how that's the stories that people tend to hear and latch onto more than, hey, I never actually got the flu. Nobody tells us that story, right? Yeah. You have to look at it in a well-controlled scientific environment. Right. So the way we test vaccines is using controls, using large numbers, patients in clinical trials. We're very, very carefully statistically analyzing side effects, benefits, costs with some kind of control where they're not getting that new experimental vaccine to see what effect that has.
I often wondered whenever I listen to people get engaged in the debate over just different kinds of vaccines, I've wondered if all of us should be responsible enough to cite statistical data. Like, for example, with the Cutter incident, which was an attenuation problem, if the real problem still was so statistically small that the benefit was far outweighed the risk. And of course, when those things happen, the system tightens up. It's kind of like it never fails when you're doing a lot of flying that all of a sudden there's a airplane disaster. And I always used to say, well, the best time to fly is like the next day because everyone's really paying attention now. But people kind of lose sight of those statistics. They kind of dwell on the exception to the rule, and it becomes, and it's frustrating because I think it's very good, and I like the way you worded it. Is that it's imperfect. We're imperfect people engaging in imperfect science, but statistically, like with polio is a great example. Despite the Cutter incident, it's phenomenal what the polio vaccine's done.
Your analogy to airfare, flying in a plane, is a really good one because when a plane crashes, it's front page headlines everywhere. Everyone hears about it, and everyone's scared to fly. Like I fly all the time. I get a little nervous when I'm getting on a plane. It just doesn't seem like this is defying physics right now when you take off in a plane. But statistically, it's safer than driving a car. Something we do every single day, mile per mile. It's something I always have to think about is in the statistical safety of something, relative to the alternative.
In the past few years, vaccines have been front page news. So we've kind of talked about that and stuff too, more than ever. What do you see as the biggest lessons learned from the COVID era vaccination efforts?
Yeah, so we all lived through that. Like, measles, typhoid, polio, that's in the history books now, but even our kids remember the COVID pandemic, and vaccines played a very important role during that pandemic. And what's interesting is there's new technology that emerged post-COVID. So that marked the emergence of what we call messenger RNA, mRNA vaccines. And this is really interesting technology. So just to back up a little bit and go back to the basic biology. So we all have this genetic material, DNA, that we inherit from our parents. That makes us who we are. So our kids have half of my DNA, and then half of my wife's DNA, and somehow combines together. Mm-hmm. And they have this now unique genome that's made of this molecule called DNA. And that DNA is transcribed in a very similar molecule called messenger RNA, mRNA. And what mRNA does, we call it a messenger because the DNA is in this very protected nucleus inside of your cells that kind of keeps the DNA safe. And then the messenger RNA takes that genetic code and it leaves the nucleus to these little protein, they're called pore complexes. And they're very carefully gated. So these little doors that let the messenger RNA out. And then the messenger RNA gives the instructions to make proteins, which is everything that we do, right? So like the ion channels that allow our neurons to communicate as we're thinking right now, right? The proteins that are allowing muscles to contract, to allow me to speak and drink my coffee, all that's powered by proteins. Proteins are what we do. And for viruses and bacteria too, right? They're making proteins that, in that case, make us sick. So what was discovered recently is rather than taking a live or attenuated virus, injecting that in hopefully a safer way to stimulate an immune response, what you can do is simply take that messenger code, a messenger RNA, and inject that into a human, and that tricks your body into making a little bit of viral protein or bacterial protein. And that will trigger an immune response. And it's much safer because you don't actually have the virus. In the case of COVID, you're not getting that COVID-19 virus into your body. You're simply getting the instruction to make one protein. It happens to be a protein on the surface of the virus that your immune system can see. It's called the spike protein. And so this is a much safer way, and it leads to much faster vaccine development, right? Because we can synthesize these messenger RNA in ways we can ramp it up. The limitation you remember about the vaccine relatively early after it was being produced, that was largely supply chain issues. That was, like we couldn't find enough glass vials and syringes, couldn't figure out how to store it and keep it cold. There's all kinds of these supply chain logistics that were being worked out. And thankfully, the government did a pretty good job, like helping support all those logistical things. You might remember Project Warp Speed, right? A lot of that was like working out all the logistics of how to, distribute this vaccine in an efficient way. And the other amazing thing about mRNA technology is you can adjust that messenger RNA to make proteins that more closely resemble that protein as the virus evolves over time, as it changes over time. And we're all aware that the original strain of COVID is very different than the strain that's circulating now. And we had all these different variants. Remember Delta and all these different variants that came through over time, and Omicron. And you can adjust that messenger RNA very quickly to produce a protein that more closely resembles the proteins on the surface of that. So it's much more effective that way. And for viruses that change quickly, like flu, like COVID, right? You have to kind of adapt your vaccine very quickly, as that virus evolves resistance to it. So it's looking more and more like COVID might be around forever, just like influenza, right? Which is bad news, but.
Well, and I remember too when the vaccine came out, so there were two, I think, companies that had like the mRNA vaccines, and then there was another company that I think had, what would we call it, like the original
the
vaccine?
Old school, that had attenuated virus.
Yeah, and there were some people that, that felt more comfortable with that type of vaccine. Why is that? Can you kind of break down maybe what some people's worries about the mRNA vaccine were?
So Pfizer and Moderna were the two companies that produced the first clinically approved in America. Other countries had their own versions of this, but the Pfizer and Moderna products were the first ones approved in the United States, and those were mRNA vaccines. And then Johnson & Johnson produced kind of an old school, like attenuated vaccine. And when they did the clinical trials of that, and they each did independently large clinical trials that involved tens of thousands of patients, where they would inject the mRNA vaccine or a placebo, and then just a saline solution that had no effect biologically. And then they would watch. These people went out into the wild. Some of them got sick, some of them didn't. They very carefully tracked the percentage of people that then got sick with COVID and the ones that didn't. And if they got sick, how sick did they get? Were they hospitalized? Did they die? Those kinds of things. So early in those clinical trials, what we saw is that the messenger RNA vaccines were about 94 to 96% effective at eliminating infection. And those that did get infected, 5-6% that did get infected, the duration was shorter. They were producing more efficient antibodies. There's a particular type of antibody that's in our secretions, like in our nose. So the virus that you then do shed is less likely to infect other people. You're sick for less time, so you're less likely to infect other people. So it made it less likely that you were going to get sick and the severity of the illness went down, but it also made it less likely that you were going to get other people sick, which is a really important, I think, discovery if we start talking about the ethics of vaccines. It's not just a personal decision. There's also public health implications, like how does it affect the people around you? But COVID-19, early in the pandemic in the United States, we saw a 0.67 case mortality rate. Like 1 in every 180 people or something like that would die of COVID-19. And we know that the older folks were much more likely to have severe cases, be hospitalized to die. And the younger folks, it wasn't too bad. Very, very small mortality rate with kids. But the vaccine dropped the hospitalization rates dramatically. It dropped the amount of cardiovascular damage that resulted from people that recovered from COVID. There was about a 50-60% decrease in mortality rate. Again, the duration of infection was shortened, the severity of infection. So lots of good things. The Johnson & Johnson vaccine was a little less effective. Still quite effective for a vaccine of this nature. It was in the 70s or something. Yeah, maybe low 80s or high 70s, something like that. So still very effective. But some people were more comfortable with that technology that had been around a while. There were a lot of rumors circulating. I remember seeing memes on social media and stuff talking about how these messenger RNA vaccines are going to reprogram your DNA and turn you into a zombie or robot or something like this, or the government's trying to control you with microchips. Being a college professor, you hear a lot of things from students, right? I think there's just that fear of something new. I think one of the most important things that I do in education is helping students understand how to weight the quality of evidence. So I spend a lot of time working with science students or pre-health students, nursing students and things like that, and talking to them about, right, here's 2 different studies that came to 2 different conclusions. Which one's better? Like the number of patients involved, how well it was controlled, this type of statistics that were used. Like we spend a lot of time talking about all that stuff. But for the general public, it's even more basic than that. Like, how do you weight a clinical study that involves tens of thousands of patients against a meme you saw on Facebook? It turns out most of the public doesn't even know what to do with those 2 things, right?
Yeah.
Or even how to access those clinical studies and start to get at that data. So I think there's an important role for education here.
And even when you get to statistics, I mean, you ask somebody, well, we've got this new thing, whether it's a vaccine or whatever, and it's 98% effective. Should we go ahead with this? And they say, oh, yeah, that sounds like really good odds. Let's do that. But if they happen to be among the 2%, or they have a loved one who's among the 2%, then they're like, no, this is awful. No, nobody should be doing this. This is dangerous. It's hard to overcome that kind of thing because, again, the 2%, like the case you were talking about with air travel and things like that, that's what gets the attention.
And also, the other problem was, it turns out, unfortunately, it's not like measles. It's not like polio. This COVID virus was evolving pretty quickly. It was changing pretty fast, which means that immunity, that really profound immunity, I mean, 95% is like about as good as we've ever seen a vaccine work, but it wasn't that durable. What happens is you take the vaccine, the levels of antibodies and therefore production build up over the course of 3-4 weeks, and you have very robust protection. Like that 95% protection was like, 4 to 12 weeks after the vaccine. But then the antibody levels start to drop off, the virus is starting to change, and the proteins on the surface of the virus start to look a little different, and your antibodies don't stick as well. And so the good news is, like, the risk in terms of death and hospitalization was durable, but the protection against infection went down over time. Or 6 to 12 months later, maybe you were only at 15% less likely to get infected than someone that was unvaccinated.
At the risk of poking the bear, there's a lot of talk today on mRNA, and the current administration has kind of pulled funding and so forth. What is their logic without us making a statement? Well, first of all, just full disclosure, I've been a fan of mRNA research from the very beginning. In fact, I remember when I reported on it to the national board, I had said that I'd always felt that it would hold the chances for a cure for the common cold. I just really like it, but I don't understand what is the blowback on it that we're getting now. Is it just anti-vax attitude, or is there some science? Or
I'm going to try really hard not to get drawn into politics. A, because I'm not a political guy. Like, I don't consider myself Republican or Democrat or, but, clearly there's political influence on these things, and that's where a lot of the funding for vaccine antibiotic research come from. A lot of people trust places like Center for Disease Control, and these are federal agencies, right? And the recommendations that they make affect, like, how insurance provides vaccines to families, the recommendations that they make. And these agencies are led by political appointees. And what's interesting is, over the years, there's been some study, like, I'll mention one in particular that I think has kind of influenced and changed the way we look at these things politically. There was a paper published in a pretty well-known scientific journal back in the late 90s. I think it was like 1998, 1999. And the author was a gentleman by the name of Dr. Wakefield, physician. He's the lead author. It was published in this journal, Lancet. And basically the conclusion of this paper was that vaccines were linked to autism. We saw this increase of autism that rose alongside the spread of vaccination around the world. And again, one of the first things we teach students in science classes is be very careful not to confuse correlation with causation, right? People buy more ice cream in the summer kind of like correlations, and then trying to figure out some causative link, like why do our taste buds change when it gets hot? Or, like you make strange conclusions if you read too much into a correlation. But that's what this was. And it turns out this paper was retracted, like it wasn't well controlled. I think there were like 12 authors, and everyone but Dr. Wakefield, who eventually lost his license, kind of said, we made some mistakes here, and like just kind of disavowed that paper over time. But that had a lasting effect on the public psyche. I mean, we've all met kids with autism. And it's true, like the diagnosis rate of autism is going up. And there's all kinds of reasons for that we could talk about, but we don't fully understand still what causes autism. And so people are trying to find something to pin that to. And that paper somehow really got out there in the media, and it kind of stuck, even though it was retracted. And now many, many, many very well-controlled studies have been done. It's showed absolutely no link. You do these placebo-controlled studies, taking vaccine or sometimes ingredients in vaccines like thimerosal, from some of these things that were like preservatives, like added to the vaccine to help give them a longer shelf life, to fix those supply chain problems, were kind of implicated in some of this too. And in many cases, those were removed. And again, they saw no difference in vaccine efficacy or safety, autism rates, things like this. And so all that's been debunked, but it still sticks. And I hear politicians kind of talking about that kind of stuff still. And it's difficult for, I think for someone that doesn't know who to trust, they see this public official talking about, hey, we're going to slow down on the vaccines, these kinds of things. And it kind of sticks with people.
And you just connected two thoughts there, or previous topics of the conversation is the COVID era stuff and the public trust of, whether it's the CDC or what your doctors are telling you or the American Medical Association or whomever, is that, it is a sad truth that the way the COVID, I'll just call it emergency, was handled here in the United States, there was a lot that was done and said that caused the general public to no longer trust the experts, whether it's people in government or people, other types of experts. And in some cases, it was people lying just because it was easier to lie than to tell the truth. In some cases, it's what you might be calling the noble lie, where they're thinking, people can't really handle this, so we'll tell them something other than the truth. But that kind of stuff, it just gets out there, and then people don't, they stop believing anything. Mm-hmm. And, now we're at a situation where the leadership of the CDC and the FDA and such has changed. There's that much more reason people have to say, well, if that just changes according to the politics, I don't think I'll believe any of it from anybody. And, that makes your job as a scientist that much harder because, you've got the data, you've got the facts, you've got the logic. But if people are saying, well, I don't feel good about that, they just won't listen.
A good example of that is the masks. Remember wearing face masks early in the pandemic, and like a surgical mask is designed and it's used in hospitals for good reason, right? I mean, there's this water-loving surface on the inside that absorbs the water droplets that are coming out of your mouth when you talk. And if physicians wear those when they're doing surgery, like droplets from their mouth that might cause an infection in the patient that they're operating on, right? Many of them get trapped. Again, masks aren't perfect, just like vaccines aren't perfect, but it really helps provide good protection for patients. We know that. There's been lots of studies done. You're less likely to have infection post-surgery if your surgeon was wearing a mask, right? And that's why that's best practice. And you wouldn't want your surgeon not to wear a mask when they do surgery on you. And we know COVID is spread through respiratory droplets. And so everyone, when this pandemic started, let's put some masks on and stay safe, right? But early there was this massive supply shortage. So suddenly hospitals ran out of masks. Nurses that are caring for COVID patients, they were having to reuse masks, and think about how gross that is, right? Horrible things like that were happening. And, well-intentioned government officials tried to stop the rush on face masks by saying, we're not recommending masks at this time, thinking we need to save them for nurses and doctors, these frontline medical workers who don't have them or can't have access to them. So the public said, all right, they're not recommending masks, they must not do any good. And then they changed their mind once the mask supply came back up. And this kind of mixed messages, we have to be very careful how we communicate that stuff, or you can lose public trust very quickly. And that's the kind of things that we saw happen during the pandemic that are just tragic in many ways.
Rob, during you had mentioned about correlation and causation. Correlation oftentimes spurs research. When people will see a correlation, it spurs it, but it's kind of like having a half of a job done. I know that we got, because we have on the CLR website, we have a paper on there on mRNA, and then we have another paper on there on the position statement on vaccines for CLR. And the people who have objected oftentimes came from a correlation perspective. And so, so the problem that you always have is you find yourself in a predicament where you don't necessarily want to disagree. Like you say, a corollary rise in autism must be vaccines, because we draw those correlations a lot in other areas, and people will automatically presume. So we've seen it before, but it's always kind of like half doing the homework. It's like you turned in your homework half done. And so when people would write me and they were angry because I wouldn't accept the correlation as concluded research, and I love correlations. I mean, I'm a problem solver, so I live by correlations, but you don't die by correlations.
You're absolutely right. Correlations, when we observe something like that, that's the reason why we set up a controlled scientific experiment, to try to get at the causative, is there a causative link between these two? That's what a good scientific experiment is. And we study those kinds of correlations because, it's interesting no matter what happens. If there's not a link between a vaccine and autism, that's important information. And if there is a link between vaccine and autism, I always try to set up experiments that are win-win that way. So no matter what happens, it's interesting, valuable data. And I encourage my students to try to find experiments like that too. But starting with that correlation is really important. But yeah, you have to do a controlled experiment with a placebo group, large numbers, carefully documenting, doing statistical analysis to see if there's a meaningful statistical difference.
Well, want to be able to talk some about the ethics of vaccine, especially as it relates to Christians as well. How can Christians think faithfully about vaccines in light of loving our neighbor and caring for the vulnerable?
Yeah, great question. So from my perspective as a Christian, humans are inestimable value, right? So we're products of divine intervention, created, we're loved by God, we're made in God's image, and all this gives us this intrinsic value. And so because of that, we have a responsibility, to protect and care for our own lives, like stewardship of our own body, but also we have a responsibility to our neighbor, right? To protect and care for the most vulnerable around us. And that's what makes bioethics surrounding vaccines complex, right? Because you have the one question of what's best to do for my body, right? On a kind of a personal level. But you also have the question of what's best to do for my neighbor. And I think one of the disheartening things that I saw during the pandemic is when students sometimes would ask me or my friends, because they know I'm a biomedical research scientist, here's the closest we have to an expert, let's ask him what he thinks. They were always asking from a personal perspective, and I always try to redirect them to think more broadly about their community. So for example, I taught nursing students, and I'd have a nursing student that, for whatever reason, was just very cautious, kind of apprehensive. This mRNA technology is new. How do we know we can trust it? I've seen the studies. I know it looks safe, but what about long-term effects? These kinds of concerns. I'd always say, it's important to think about those things, right? And those are legitimate concerns. But it's also important to think about your patients, right? You're scheduled to do a rotation at Children's Hospital Wisconsin next semester. Most of the kids that are there, a lot of them have leukemia, which is impacting their immune system. And we know that this vaccine reduces the likelihood that you're going to make someone else sick. Not only you get sick, but the nasty thing about COVID is sometimes you can even have asymptomatic transmission. Especially in young people, like students, they can get just a little bit sick and then spread that virus without realizing it. It would be much simpler if, you had a runny nose, stayed home, it wouldn't spread. That wasn't the case with COVID, right? You were shedding infectious virus even before all the symptoms came on sometimes. So by taking that vaccine, you're not only making you safer, you're making your grandma safer. When you visit her, you're less likely to infect her. You're making those kids with leukemia at Children's Hospital safer. There's a lot of things to kind of weigh and balance there, and it becomes really complex. Another factor, I think, we looked at back statistically, some of the groups that were more likely to not accept the vaccine during COVID were Protestants and Catholics. And I think a big part of that was work that was done on the research and development side to develop the COVID vaccine, understand how it works. I mentioned before, and I was talking about Jonas Salk and how he was growing viruses in petri dishes with cells. Well, in some of the research and development of making vaccines, sometimes we use human cell lines. And some of those cell lines have interesting paths and sometimes bioethically complex paths. So, for example, I think it was the Pfizer line. There's some research and development work using a cell line called HEK293 cells, HEK293. And HEK stands for human embryonic kidney cells. And what most people, like my students, when they encounter these in the lab, it's funny because I'll get emails from a student that I taught 10 years ago, like, hey, Dr. Balza, I'm using these cells. I just looked them up. HEK293, I didn't even think about what they were. Just culturing them. I actually looked them up on Wikipedia or something, and they're human embryonic kidney cells. Can you tell me about the bioethical implications and things like this? And these cells are really interesting. They have a long past. So they're about as old as I am. They were developed in the mid-1970s in the Netherlands. And we don't really know, like, they came from human embryonic kidneys, but we don't know if it was that, a miscarriage, or was that an elective abortion, or what happened, like that history. We don't know anything about that. So there's a possibility that there was an elective abortion. Some of these cells were biopsied, placed into culture, and propagated in labs, and then passed down over the years. And so for some 50 years, these cells have been propagated, and we're using them to research things like vaccines and many other things, by the way. Just to be clear, the vast majority of the US pharmacopoeia is developed using cell lines that ultimately at some point in their past were developed from fetal cells. And so the question that a lot of people wrestle with is, like some of my students would wrestle with, am I morally culpable if I'm working in a lab that uses these cells in abortion? Or thinking about it from a patient perspective, if I accept a vaccine that at some point in the development of the technology used to make that vaccine, they were tested on these human cell lines, am I morally culpable in that abortion in some sense? And these are good questions to be thinking about, and things that I wrestle with too. What I'll say to that, though, is I think you have to think about several things. Think about timing, proximity, knowledge, intent, weigh all these factors when you're thinking about. Here's an example. You can just kind of think through this complex problem. I think if you're pro-life, as I think we all are, we would all agree that it would be immoral to be a surgeon performing elective abortions in a clinic. We'd also probably agree that if there was a nurse there assisting that physician, handing her tools or something, right, that nurse is probably morally culpable in that abortion as well. But what about the janitor that takes out the trash from the abortion clinic? What about the secretary at the abortion clinic? What about the bus driver that brought the secretary to the abortion clinic that morning? Is that bus driver morally culpable in that abortion? What about the gas station attendant that pumped gas into the bus that took the secretary that scheduled the abortion, right? So you can keep going back further and further and further. And if you think about it, like everything we do, like touches sin in some way, right? I heard somebody make the argument that, you shouldn't buy a Volkswagen because they built machines for the Nazis back in World War II, right? Are you morally culpable for the Holocaust if you're driving a Volkswagen, right? Those kinds of arguments, like how far back do we take this, right? And if so, like, can we do anything? Can you buy any product that's not produced in some sort of sinful way? And so it's really hard. And so weighing things like that against protecting your neighbor, protecting your mother. You might have a spouse that's dealing with cancer, going through chemotherapy, and or has lung disease already, and is very susceptible to COVID. These are complex moral decisions, right? And from a Christian perspective, obviously, like following one's conscience on these matters is very, very important. But also to having a clear understanding of, like, you hear people say things like, well, you're just injecting fetal tissue into your body when you accept this vaccine. That's just not true. Every time, they develop a vaccine, they abort babies. That's also not true. In fact, you can make an argument these cell lines reduce the demand for fetal tissue because they're immortal, right? These HEK293 cells have been growing for 50 years, and they can grow for 500 more years. We'll never run out of them. And therefore, in some ways, like the use of these cell lines reduces the need for experimental material derived from an abortion or something like that. So really complex issues. And kind of sometimes the more you think about it, the more confusing it gets in some ways, but it's important to think through these things.
Just one final question. If you could leave our listeners with one guiding principle for navigating vaccines wisely and faithfully, what would it be?
Oh, great question. I'm a Lutheran, and so I'll say from a Lutheran perspective, Luther always talked about God providing for us through masks. the
not surgical masks.
Not surgical masks, the mask of God, Luther would often talk about, right? So in other words, like God provides for our daily sustenance through butchers and bakers and farmers. And these are the masks through which he provides for us, right? And so some people have told me, like, for a Christian to accept a vaccine demonstrates a lack of trust in God's providence. And I would say, if you think through that from a Lutheran perspective, that vaccine may be God's providence, may be his way of protecting you, your loved ones, your community for something like this. So we're not just the product of God's providence, we're also the agents of God's providence. And I teach my students that are going into medicine or science this too, right? God is going to serve your community through you in these fields, much like the way, God provides for a congregation through the pastor. Lutheran idea of vocation. And I think that's a concept that gets lost a lot in these discussions. So I guess I just leave my Christian and Lutheran friends that are wrestling with these topics with that idea, to kind of think about too in the midst of all this.
If I can just add on to that, the vocation of the average person is also that of a citizen. And as Christian citizens, we have an obligation to love those around us and look out for what's best for them. And that's, again, the previous things, whether or not we get vaccinated or have our kids vaccinated, that is something that affects other people as well.
Right. Well, thank you very much for joining us. And we really appreciate it. And for all of our listeners, too, if you have any questions on this topic, you can reach us at lifechallenges.us. Thanks a lot, and we'll see you back next time. Bye.
Thank you.
Thank you for joining us for the Life Challenges podcast from Christian Life Resources. Please consider subscribing to this podcast, giving us a review wherever you access it, and sharing it with friends. We're here to help, so if you have questions on today's topic or other life issues, you can submit them, as well as comments or suggestions for future episodes at lifechallenges.us or email us at podcast@christianliferesources.com. You can find past episodes and other valuable information at lifechallenges.us, so please check it out. For more about our parent organization, please visit christianliferesources.com. May God give you wisdom, love, strength, and peace in Christ for every life challenge.